Hello everyone, my name is 𝙂𝙋𝘼𝙎. I hope you're doing well. Let's move towards the next topic, that is autonomic nervous system, cholinergic system. In this video, we're going to learn what is cholinergic system. First part, until the pharmacological actions of acetylcholine. Okay, but before that, we need to discuss the previous question, what we have for you in the last video. So the question was, the which structure act as a major integration center for autonomic functions? And the next question is, the dual innervation by sympathetic and parasympathetic system is absent in which of the following organs? Heart, eye, blood vessels, or lungs? So let's see the answer. So the answer is, which structure act as a major integration center for autonomic system is hypothalamus. Through hypothalamus only you will get the autonomic functions. Okay. Then dual innervation by sympathetic and parasympathetic system is absent in. So except blood vessels, many of the organs have both the innervations, parasympathetic also as well as sympathetic also. But in the case of blood vessels, sympathetic system does not work. Only parasympathetic conditions works. Okay. So now let's move towards today's topic, that is cholinergic system. So what we have in this video, we have the introduction to cholinergic system. Receptors we're going to discuss, muscarinic as well as nicotinic. Synthesis, storage and destruction of acetylcholine. This one is very important. Pharmacological actions of acetylcholine on different organs. Okay. And then important questions as we always do. Now, so acetylcholine is the principal neurotransmitter in the cholinergic system. As I have told you earlier also, acetylcholine will be the principal neurotransmitter in cholinergic system. Okay. Then the neuron that synthesizes from a storage and a release acetylcholine are called cholinergic neurons. So the neurons which are responsible for the storage and the release of acetylcholine, those are the cholinergic neuron. Okay. And also acetylcholine releases from the cholinergic nerve ending. As we have discussed before, there is a nerve ending. So acetylcholine synthesized from there only. Also, the acetylcholine is a quaternary ammonium compound. So it is a quaternary ammonium compound. So what will be its nature? Is it lipophilic or hydrophilic? Is it lipid soluble or water soluble? It will be a water soluble. So if it is a water soluble, then what kind of decomposition can be take place? Hydrolysis. So that's why it can undergo rapid hydrolyzation by cholinesterases. The enzymes which are responsible for the decomposition of acetylcholine are the cholinesterases. So it goes under rapid hydrolyzation, hence it has no therapeutic applications. Okay, so you have learned the cholinergic system. What is cholinergic system? Cholinergic system is basically the system which initiates the parasympathetic conditions in our body. The principal neurotransmitter of cholinergic system is acetylcholine. It forms through the cholinergic neuron. Also, it is a quaternary ammonium compound and gets hydrolyzed by acetylcholinesterases. Acetylcholinesterases, butyrylcholinesterases. There are two enzymes, acetylcholinesterases and butyrylcholinesterases, which are responsible for the decomposition of acetylcholine. Okay. So now let's move towards the cholinergic receptors. So what are the receptors through which the acetylcholine will bind? So there are two types, nicotinic and muscarinic. In all the types of cholinergic receptor, acetylcholine will be bind. Okay. So let's see the locations of these types of receptors. So first, before knowing the locations of these types of receptors, we need to know what is the nature of these receptors. Okay. So nicotinic receptors are ligand-gated ion channel. There is a ligand which undergoes through that channel and causes depolarization and activation of that receptor. But if we talk about the muscarinic receptors, these muscarinic receptors can be work through G coupled receptors. Okay, G protein coupled receptors. And there is also a variation in between them. M1, M3, and M5, the odd ones, will be the GQ coupled receptors. And what will be the down signaling? Down signal will undergo through guanylyl cyclase, which is a primary messenger. Then downwards, IP3 DAG pathway. IP3 DAG pathway, calcium will be released and then goes towards the contraction. Okay. So GQ is responsible for the contraction, whereas the even ones, M2 and M4, are responsible for the relaxation. Okay, the relaxation. Why it is? Why is it so? Because the M2 and M4 are GI coupled receptors. Okay, so the GI coupled receptor undergoes through M2 and M4 receptors. Okay, so now let's move towards the location of these types of receptors. So NN is one of the type of nicotinic receptor which are located into the autonomic ganglia, adrenal medulla. Okay, autonomic ganglia and adrenal medulla. NM is NM, neuromuscular. NM can be learned through neuromuscular junction, okay, between the skeletal muscles. Now, muscarinic receptors, gastric ganglia, gastric, okay, GIT, and then CNS M1, okay. Next, M2 is for heart, M3 is for eye, GIT, bladder, bronchus, gland, CNS. So they are particularly for the smooth muscles, okay. Smooth muscles in GIT also, bladder also, bronchus also, glands also, okay. Then CNS and CNS. So CNS will be common in all the types of muscarinic receptors, but what do we have to learn the other types of sites? How do we learn? So you can learn from a trick that first eat, then gastric. Okay, eat will be correlated through gastric. Okay, then first eat and then fall in love. First eat, then fall in love and rest and do rest of the things far ahead. Okay, then rest of the things far ahead. So I can write here also that first eat, then fall in love, and then rest of the things far ahead. Okay. First eat, fall in love, and then rest of the things are far ahead. So first eat, you can correlate it through gastric ganglia. Then fall in love will be through heart or brain also. Okay. Then heart and brain. And rest of the things will be in the M3. Okay. So these three major receptors should be learned by you because most of the pharmacological actions have these three receptors only. M4 and M5 will be only the CNS ones. Okay. Let's move towards the next slide. So nicotinic receptors. So what are the nicotinic receptors? As we have told you that nicotinic receptors are expressed by the ligand-gated ion channel. Activation causes by opening of ion channel and the depolarization. So what is the depolarization? Whenever there is a increased amount of positive ion inside the cell, that is called as the depolarization. Okay. Then these types of nicotinic receptors are divided into two types, NN and NM. NN is for autonomic ganglia and the adrenal medulla, which is stimulated by DMPP, dimethylphenylpiperazinium, which is also blocked by hexamethonium, This is very important to know what is the selective agonist and what is the selective antagonist for NN or NM receptors. Okay. Now you can learn it through uh, drive heartbeat with nerves. Drive heartbeat with nerves. Drive for dimethylphenylpiperazinium. Then heartbeat hexamethonium with nerves. Nerve will be signify for NN. Okay. So drive heartbeat with nerves. Okay, let's move towards the NM. So NM is for the contraction of the neuromuscular junctions. Okay. Then it is for skeletal muscles, which is stimulated by phenyl trimethyl ammonium. That is phenyl trimethyl ammonium, and it is selectively blocked by the tubercular. Okay. Now it can be learned through pure throttle for muscle. Pure throttle for muscle. Pure for phenyltrimethylene, throttle for tubercularine, and for muscle NM receptors. Okay. So this is clear that, and it is very important to learn also that what are the selective agonists and what are the selective antagonists for both of these receptors. Okay, stimulated by phenyl trimethylammonium, blocked by tribocurarine for NM receptors. In the case of NN, dimethyl phenylpiperazinium is is used as a stimulated agonist for NN receptor, and it is selectively blocked by hexamethonium. Okay, so you can learn these things from this trick. Now move towards the muscarinic receptor. The muscarinic receptor, I have already told you that it is a G protein coupled receptors. Okay, and G protein coupled receptors, which are basically GQ mediated as well as GI mediated. So the odd ones will be the G1, G3, and G5. These three, G1, G3, and G5, will be GQ mediated, whereas M2 and M5, M2 and M4 will be GI mediated. Okay, M1, M3, and M5 will be GQ mediated. M2 and M4 will be GI mediated. Okay. Now, it will be selectively blocked by atropine, not selectively, non-selectively, it will be blocked by atropine. All the types of muscarinic receptor will be blocked by atropine, whether it is M1, whether it is M2, M3, M4, or M5. Now, let's move towards the selective agonist as well as the selective antagonist. This is very important to know. Okay, for M1 receptors, so where it is belong, M1 is for the gastric glands. Okay, first eat, then fall in love, and then rest of the things can wait or far ahead. Then let's learn the selective agonist. Selective agonist for M1 is oxotrimorin, whereas the selective antagonist is telanzapine and paranzapine. So it can be learned through the OTP, 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 OTP for M1. Okay, before you eat, or after you eat, you're gonna pay the bill. You're gonna pay the bill through your debit card, maybe, and then afterward you will get the OTP, isn't it? OTP on the mobile. So that's why I have written OTP for M1 receptor to eat after eating. Okay. Then M2 is for heart, fall in love. So M2 is for myocardium. M2 is for myocardium. M for myocardium. M for methacholine. Okay. Then metoctramine and tryptamine is for the selective antagonist of M2, and it can be learned through MMT. Then M3 is selectively for the bladder. Okay, bladder. The bladder, B for Bethany-Gall. B for bladder, B for Bethany-Gall. And then antagonists are the deraphenecin and solifenacin, that can be learned through PDS. Okay. Now let's move towards the most important topic of this class, that is synthesis, storage, and destruction of acetylcholine. Okay. So now let's focus here. So this is a cholinergic axon. Okay. So this is a cholinergic axon in which the acetylcholine synthesizes. So it is synthesizes through one enzyme, that is choline acetyl transferase, that is CHAT, choline acetyl transferase. Okay, which catalyzes the synthesis of acetylcholine from choline and acetyl CoA. So what is the amino acid which are responsible for the synthesis of acetylcholine? That will be choline as well as the acetylcholine. That should be remembered by you, that choline and acetyl coenzyme A, choline and acetyl coenzyme A are responsible for the synthesis of acetylcholine. And which enzyme responsible for it? Because it catalyzes the synthesis of acetylcholine. And what is the name of that enzyme? That is choline acetyl transferase, that is CHAT. Okay. Now, choline gets into the cholinergic axonal membrane, and then acetyl coenzyme A also. From this, choline acetyl coenzyme A and then CHAT will catalyzes the reaction, and it will form the acetylcholine. Okay. Now, the acetylcholine will be stored in the synaptic vessels. It will store under the synaptic vessels. So there is a uptake into storage vesicles. So this acetyl, for why it is going to be stored in the storage vesicles? Because for the protection, acetylcholine is protected from degradation in the vesicle. Under the storage, you will not be able to meet out the any other foreign substances. Okay, so that's why acetylcholine will be stored into the storage vesicles. Okay, then after its storage, it can be released also. So synaptic vesicles release this acetylcholine for its pharmacological action. How it will show its pharmacological action? By binding onto the receptors. So receptors on the postsynaptic side. Now, so it should be released from this, from here. So then next step is to release the neurotransmitter. So release is blocked by botulinum toxin. Botulinum toxin is a toxin which block its release from the synaptic vessels. Here also, 𝘂𝙾𝘾 is a drug which inhibits the uptake in storage vesicles. These are very important to understand that 𝘂𝙾𝘾 is a drug that which inhibits the uptake into storage vesicles of acetylcholine. Okay, so release will be inhibited by the botulinum toxin and the storage vesicle VES for 𝘂𝙾𝘾. VES for 𝘂𝙾𝘾 inhibits the acetylcholine. Okay, now so when it releases into the synaptic cleft, now what happens? There is a lots of what? Acetylcholinestrases, butyrylcholinestrases, which are responsible for the decomposition, hydrolysis of acetylcholine. Okay, so what happened towards the acetylcholine best? It goes into the decomposition or hydrolysis by acetylcholinestrases and butyrylcholinestrases. It gets hydrolyzed again into choline. It gets hydrolyzed again into choline, and this choline is get reuptake into the axonal membrane. Okay, that is also called as the recycling of choline. Okay, so here choline is being recycled into the axonal membrane, and then what happened? This is a rate limiting step also, because it is a slow step and it is also a rate limiting step. Here the drug hemicolinium, that is hemicolinium, inhibits this recycling of choline. Okay, choline is taken by the neuron. This transport is inhibited by the hemicolinium. Okay, so this is very important and is asked in various of exams that what does hemicolinium do in the synthesis, storage, and destruction of acetylcholine? So it generally blocks the choline uptake into the axonal axonal membrane. Okay, so now acetylcholine is now being hydrolyzed into choline part and the acetate part. But if it does not uh, decomposed or hydrolyzed into the choline and acetate part, so what will exactly do? It will exactly do the binding onto the receptor, binding onto the nicotinic receptor or by the muscarinic receptor. Then afterwards, whenever they bind into the muscarinic receptor or into the nicotinic receptor, these nicotinic receptor as well as the muscarinic receptor have their own pharmacological actions downwards. That pharmacological actions we will learn into the next slides. Okay, so let me concise it into the very short part that choline and the acetyl coenzyme A is responsible for the acetylcholine synthesis, whereas CH80, that is choline acetyl transfer, is responsible for the catalyzation of this reaction. Now it goes into the storage vesicle where where it is being protected. But this storage can be inhibited by 𝘂𝙾𝘾. Now it gets released into the synaptic left. Here also the release can be inhibited by the neurotransmitter. Sorry, the release can be inhibited by the botulinum toxin. Okay. Now, when it reaches into the synaptic left, there are two scenarios: whether it can hydrolyze into the choline or acetate part, or it bind towards the receptors. So when it degrades or hydrolyzes into the synaptic left, it hydrolyzes into choline and acetate. This choline gets reuptake into the axonal membrane, and this reuptake can be inhibited by the hemicolinium, which is a rate limiting step. Okay, so this should be remembered by you that blocks choline uptake, hemicolinium, blocks vesicular uptake, Westmacol, block release of acetylcholine, that is botulinum toxin. Now we're going to discuss the pharmacological actions of acetylcholine. So now we will see the pharmacological action of acetylcholine. So now the acetylcholine produces muscarinic and nicotinic effect by interacting with their respective receptors on their effector cells. As I've told you, the acetylcholine will bind towards the nicotinic receptor as well as the muscarinic receptors. So both the receptors have their own pharmacological actions. Okay. So now let's see the muscarinic actions. Okay. So as we have discussed, the cardiovascular system is come under the M2 receptor. No, first eat, then fall in love, and rest of the things will be wait. Okay. Now, the acetylcholine act by stimulating M2 receptor of the heart, opening potassium channel, resulting in hyperpolarization. Okay. So, what will acetylcholine do? Acetylcholine will do the binding onto the M2 receptor, and then it will open the potassium channel, which will are responsible for the hyperpolarization. So hyperpolarization means what? The potassium channel opening with the potassium which is inside the cell, it will go outside the cell. That is called the hyperpolarization. Okay. So now what will it do? The acetylcholine act on the M2 receptor and then open the potassium channel and resulting in hyperpolarization. So what is the purpose of hyperpolarization is here? So hyperpolarization will result into the down in heart rate, force of contraction, and the down in AV conduction. Okay. So here heart rate will be decreased, contractility will be increased, and the conductivity will also be decreased. So as we have discussed before also that on the heart, acetylcholine has a relaxation property. Okay, although acetylcholine is responsible for the relaxation on the heart, whereas sympathetic system, whether what are the adrenergic drugs, dopamine, noradrenaline, adrenaline, these are responsible for the exaggeration, activation of the heart rate or increase in the heart rate, force of contraction, atrioventricular conduction. Okay. So, in the heart system, in the cardiovascular system, these um, acetylcholine will result in the relaxation, and in the case of sympathetic, it will cause the contraction, more contraction of heart. Okay. Now, also, acetylcholine acts on the M3 receptor on blood vessels. So blood in the blood vessels, M3 receptors can be found. So here, acetylcholine are responsible for the vasodilation because of the release of NO, that is nitric oxide, not nitrous oxide. That will be N2O. NO is nitric oxide, which is a endothelial relaxing factor, endothelial dependent relaxing factor, which is responsible for the down in BP. Okay. So now let's move towards the smooth muscles. So smooth muscles, we have the gastrointestinal tract. So M3 are responsible for the defecation purpose in GIT. Okay. So now the in the gastrointestinal part, acetylcholine, because it has a relaxation property on all the other organs except no, acetylcholine has excitatory actions on all the other organs except heart. It will relax heart but increase all the tone secretion on all the activity of other organs. So here if we talk about the gastrointestinal tract where defecation processes may be taken, so relaxes the sphincter, which are there is a sphincter. If it is contract, so defecation process will not be allowed. No. So the content will not be able to flow. Okay. Then how it will be occur. So because it increases the tone of the gut, it will decrease the tone of the gut. That is a tension in the smooth muscles. Also, it will increase the peristaltic movement of intestine. Also, the GI secretion will also be increased. That is responsible for the relaxation of sphincter, opening of the sphincter. Sphincter is just like a valve. Sphincter is just like a valve or the opening of tube, anything of that. Then, due to the relaxation of sphincter, all the material will flow away. That is responsible for the defecation. Next, urinary bladder. So there are two types of muscles in the urinary bladder. First one is detrusor muscle and second one is the sphincter muscle. Sphincter muscle is the last part where through urine passes out. Okay. So now the acetylcholine acts on the M3 receptors here contracts the detrusor muscle, the upper part. The upper part is the detrusor muscles, whereas the second one part, the lower part, is the trigone and sphincter muscles. Okay, so sphincter is just like a wall, as I have told you before also, that sphincter is just like a valve. When the valve gets open, the defecation or the urination process will start. It will cause the urination in the case of urinary bladder. So it will contract from the upper side and then relax from the lower side, which will result into the urination. Now relax. Now let's move towards the bronchi. So bronchi, because of the M3 receptor, it will cause what? It will cause the GQ, it will cause the contraction. Why contraction? Because it is a GQ mediated. No, it is GQ mediated. So it contracts the bronchial smooth muscles. The bronchospasm may take place. Increased tracheobronchial secretion. Therefore, cholinergic drugs are contraindicated in asthma. So what generally happens in asthma? Asthma is the condition where bronchospasm or the bronchoconstriction take place. The bronchioles, the diameter of bronchiole gets constricted. But if we talk about the asthmatic condition, in asthmatic condition, bronchoconstriction is already there. But if we take the acetylcholine or any synthetic analog of acetylcholine into the asthmatic condition, it will exaggerate the bronchoconstriction. No, it will exaggerate the bronchoconstriction. So that's why these types of cholinergic drugs, are contraindicated in asthma conditions. Next, we should talk about the glands. So glands also come under the M3 receptors, which are the GQ, no, which are responsible for the increase in glandular secretions. Glandular secretions are responsible for the HCl secretion, saliva, and lacrimations. So, in the previous lecture, also I have told you one thing, that is SLUD, 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 S for salivation, L for lacrimation, U for urination, D for defecation. Also, the HCl secretion will be increased in the parasympathetic conditions. Now, let's talk about the male sex organ. Due to vasodilation in cholinergic system, it is responsible for erection of the male organ. Okay, so whenever there is a increased blood flow inside the penis, it is responsible for the erection. So what is the science between the erection? So erection occurs whenever you are in the parasympathetic mode, in the relaxed mode, and then in the relaxed mode the blood flow increases inside your penis, and that increase in the blood flow are responsible for the erection of male sex organ. Okay, now let's move towards the pharmacological action of acetylcholine. So acetylcholine in the nicotinic receptors, that is, we are going to learn the nicotinic actions. Now in the autonomic ganglia, both parasympathetic and sympathetic ganglia are stimulated by acetylcholine through the stimulation of NN receptors. Here, sympathetic ganglia as well as the parasympathetic ganglia, both are stimulated by acetylcholine through the NN receptors. Now, neuromuscular junctions, these are responsible for the mobility in skeletal muscles. So, acetylcholine stimulates skeletal muscles contraction on NM receptor. So whenever it binds through NM receptor, which is a ligand-gated ion channel, both are the ligand-gated ion channel, NM receptor or NM receptor. So when acetylcholine acts on the NM receptor and downstream signaling which passes towards the ligand-gated channel, it causes the depolarization. Depolarization are result into the contraction of skeletal muscles, which will be occur on the neuromuscular junctions. So this is all about the video. Now we should discuss some important questions and wind up this class. So first question is acetylcholine is synthesized from which of these? Okay. The second question is acetylcholine causes bradycardia by acting on. So you have 10 seconds to answer. Okay. So the answer is the acetylcholine is synthesized from choline and acetyl coenzyme A. As we have told you before that choline and acetyl coenzyme A are the two chemicals which are responsible for the acetylcholine synthesis. Whereas CHAT, that is choline acetyl transferase. Choline acetyl transferase is an enzyme which are responsible for the catalysis of this reaction. It catalyzes this reaction. Also, the acetylcholine bradycardia. Bradycardia means decrease in the heart rate. So, from that trick, you can decode this question that acetylcholine causes bradycardia through M2 receptors. First, eat, then fall in love with your heart and brain. Heart will come onto the M2 receptors. Okay. Now, so next question: which of the following is a ganglionic blocker? So here is a selective ganglionic blocker. It is being asked in this question. Next question is which drugs inhibits choline uptake into the presynaptic terminal? You have 10 seconds to answer. Let's see the answer. So which of the following is a ganglionic blocker? Ganglionic blocker is a selective antagonist of NN receptor, that is hexamethonium. Okay. Autonomic ganglia is being discussed over here. Ganglionic blocker. Okay. Autonomic ganglia is a ganglionic. So the what is the blocker? What is the selective antagonist of it? It is hexamethonium. Okay. So which drug inhibits choline uptake into the presynaptic terminal? Hemicholinum, which is a rate limiting step. Okay. Now, which of the following statement about muscarinic receptor is true? So you have to tell me the right statement among these. Okay. Now, which drug prevents acetylcholine storage in vesicles? This is a very easy question. 10 seconds. So the first statement is these are ligand-gated ion channel low. What are the ligand-gated ion channels? NM and NN receptors. They are blocked by nicotine? No, it is not blocked by nicotine. They are G protein coupled receptor? Yes. All the muscarinic receptors are G protein coupled receptors. Okay. Which drug prevents acetylcholine storage in vesicles? Hemicholinum, vesamicol, botulinum toxin, or physostigmine. So vesamicol, I have make you learn the vesicles can be correlated to the vesamicol. So these are the answers. They are a G protein coupled receptor and the vesamicol is responsible for the prevention of acetylcholine storage in vesicles. Okay. Next. Nicotinic receptor are located all of the following except which muscarinic receptor subtype is inhibitory in function? Okay, so these types of questions are already being discussed in this class only. So it will make you revise also. Answer. Let's see the answer now. Nicotinic receptor are located all of the following except autonomic ganglia. Yes, it is present over there. Skeletal neuromuscular junction. Yes, it is present over there. Adrenal medulla. Yes. In smooth muscles, no. Muscarinic receptor are located into the smooth muscles, the M3 ones. M3 receptor are responsible for the location in smooth muscles. Which muscarinic receptor is inhibitory in function? M2 receptor. I've told you that odd ones, M1, M3, and M5. These three, M1, M3, and M5 are excitatory in action, whereas M2 and M4, the even ones, are inhibitory in action. So now these are the questions for you, which you have to tell me in the comment box, and we'll discuss into the next class. So which one of the following is a false statement about acetylcholine? Which is one of the following is a false statement? You have to tell me the wrong statement among these. Next question is the primary mechanism for termination of acetylcholine is. So what is the primary mechanism for termination of acetylcholine is? No, this is not the answer. So let me delete this question. So you have to answer this question only. Okay, which on the following is a false statement about acetylcholine? First option is it causes vasodilation via nitric oxide. It is not used therapeutically due to rapid degradation. It crosses the blood-brain barrier easily. It causes contraction of nitrosomosis. So this is all about the video. I hope you like it. I hope you um, make it understand. I make you understand. And if you like the video, please do like this video onto the YouTube symbol, and we will meet in the next class. Until then, tata, bye bye.